All right, so we're going to talk about antibiotics and chemical control today. Basically just ways to control bacterial growth. And we can do it in a number of different ways. There's things like disinfectants, obviously, but we can also use antibiotics or other types of chemicals that slow bacterial growth. I know Dr. Lawson likes this video. You guys have seen this video before, right? Yeah, okay, I won't show you again. There's just one that I like that demonstrates really quickly and effectively how small mutations evolve rather quickly for antibiotic resistance. And just because you have something that's susceptible, eventually can become resistant pretty quickly, depending on the organism. A couple of fun facts that usually isn't discussed about this particular video. They talk about this big plate. The plate is actually about the size of this table right here. It's actually really cool. And one of the ways that they made it, they don't talk about, is they used squid ink and India ink, which are things that bacteria don't really care about, that doesn't harm them. So they have to make the background black, so most media obviously isn't black, it's see-through, so they put it with India ink to make it really good at contrasting when they took the camera and stuff. It was actually really interesting. But the whole video, very, very effective way of showing how resistance is developed. So we're gonna talk a little bit about how antibiotics work. Now, antibiotics are one of my favorite things, but they do get very complicated. So if there's something that you don't understand or you're kind of confused about something, by all means, definitely ask for help, or if you're not sure how something works. Generally speaking, the things that you need to know about the bacteria, when we talk about an antibiotic, is how it works. What is its mode of action? How is it killing things? How is it interfering with the metabolism? What is it doing to the bacteria? Also, you should know whether or not it's bactericidal or bacteriostatic. Bactericidal means what is it killing the organism? If it works by killing the organism, then it's bactericidal. If it just slows bacterial growth, it's considered bacteriostatic. So it just slows everything down. And you might think to yourself, hey, why don't we just make everything bactericidal? Well, those drugs have a tendency to be a little bit more high risk for us to use and have higher side effects, whereas bacteriostatic generally tend to be a lot easier for us to use with fewer side effects we can take a lot more of. Now, this isn't always the case, but generally speaking, we want our immune system to be able to handle the bulk of it and not have to rely on it getting wiped out by the drugs that we take. So having our immune system work efficiently is a good goal to have in this. So the next up thing that we're talking about is therapeutic index. So what is a therapeutic index? Therapeutic index is a really tiny math equation, and it's actually really easy. It's basically toxic dose divided by therapeutic dose. The toxic dose is just how much medicine do you have to take before you see undesirable side effects. Therapeutic dose is how much do you have to take of a certain drug or medicine before you get the desired effect. And then you divide the toxic dose by the therapeutic dose, and you figure out what the therapeutic index is. The larger the therapeutic index is, means that the more of that drug you can take without getting negative side effects or severe side effects. So a great example of this is ampicillin, which has a therapeutic index of 100. So that means for a therapeutic dose, a toxic dose would be 100 times that, which is more than a human could conceivably consume at once. So if you think about a normal dose of ampicillin, 50 milligrams of ampicillin, what have you, you would have to take 100 times that. Well, that's a lot. You probably couldn't consume that. That's even more than the total number of pills in the bottle. So it's actually relatively safe. You couldn't really hurt yourself with ampicillin much unless you were allergic to it, which we'll talk more about in a second. Something like gentamicin, though, has a very low therapeutic index. It's a therapeutic index of 3. That means you only have to take a little tiny bit about it, and if you take a little bit more than that, then you get some negative side effects. That's not as good of a drug. It's not as safe. And part of this is kind of a sliding scale thing. Sometimes you build up drugs over the lifetime of it or taking it. So if you take a whole bunch of it for a long period of time, you start getting the buildup, and that buildup starts leading to those side effects. A really common and great example. So a great example of this is actually, if you guys remember when we were first having COVID out, there were some papers released about the efficacy of hydroxychloroquine. Which is used to treat lupus and some other things. And we got a whole bunch of different things that we can use it for. The problem with one of the things that was reported is that news people would report on it and they'd go like, hey, you shouldn't be taking this. One of its side effects is that it can cause you to go blind. was like, well, yeah, that is true. It does cause degradation of vision. That is a known side effect. But the trick is that that side effect is only seen in patients who take that long-term for multiple years in a row at high doses. So the minimum threshold is 5 years on the medicine, accumulation of 1 kilogram total of consuming that medicine. A kilogram of medicine. And that's a really long time. And even when you get to that point, the percentage of patients that actually have vision loss at 5 years is really low. It's like 5%. And then you have to get 10 years of taking it before you start really seeing an increase in the number of people who have it. So if you think about it, that's like a huge thing. So yes, you can have a side effect of blindness from it, but that side effect isn't an immediate side effect. You have to take a huge amount of that drug for a really long time to start getting that side effect, not over the course of a viral infection. And of course, doesn't seem to work very effectively, so on and so forth, for COVID, but that's neither here nor there. It was just kind of a weird, hey, there's this dangerous side effect to it. Well, yes and no. So it's always important to know how those side effects work. Now, some side effects, some therapeutic indexes are really low, and sometimes the therapeutic index is just a 1 or less than a 1, because you are always going to get negative, undesired side effects at any dose, even below a therapeutic dose. Some examples of this are antifungals. Some of them are very, very dangerous, and you can take them and they can actually just shut down your liver. And in part that has to do with the way that eukaryotic cells are very similar to each other. Since fungi are eukaryotes and our cells are eukaryotes, something that affects them has a high likelihood of causing us problems. Same thing with cancer treatments. Because cancer cells are just our cells going kind of crazy, some things that will affect them really negatively will also affect some of our other cells and cause those side effects. So that's an unfortunate reality of some of these things. Okay, so we're going to talk about classes of antibiotics. There are a bunch of antibiotics, but they tend to fall into major classes. Now, these classes aren't comprehensive because there's like divisions within each of these classes. We're going to cover these kind of major ones. And a great example of this is penicillins, which include things like penicillin, amoxicillin, ampicillin, and what have you. Those are the kind of common examples. But there's a type of penicillin drug that's really commonly prescribed that is a penicillin, but it's a little bit different, and so it's got its own little class. Does anybody know what that class is? It's a really common one. Put in a lot of antibiotic ointments. So cephalosporins. So those are types of penicillins, but they're kind of different enough that they're in their own little class. So just keep that in mind. So this isn't comprehensive. This is just kind of like large groupings that are easy for us to talk about. Okay, great. Sulfa drugs. These are sulfonamides, which include ones like sulfasoxazole, which I know rolls right off the tongue, but that's the really common one. There's also things like SXT or Bactrim. Bacteriostatic, so it only inhibits the growth of bacteria, it slows it down. And the way it does this is through competitive inhibition of folic acid synthesis. So it's an antigen metabolite, it competes with PABA during folic acid synthesis, and we need folic acid to make things like amino acids and purines. But we don't necessarily have to have that, especially if we can just take it from other resources. So we don't technically have to have a lot of folic acid synthesis. We can just consume folic acid or consume the amino acids or the purines, and we get it. So we don't have to have that pathway. The pathway for that production, we don't use it. So we don't care if you're inhibiting with PABA for humans, it doesn't hurt us. But for bacteria, it's kind of a big issue. So it works really well on them. The side effects of this are, generally speaking, fall under the category of allergic reaction. That's the most common one. I'm actually allergic to sulfa drugs, which annoys a lot of doctors because it's a really convenient one because you can give a huge amount of it and the likelihood of you having some sort of bad reaction is really low. And it's considered broad spectrum, so that means it works on a bunch of different types of organisms, primarily used for gram-negatives, but it can work on gram-positives too. And so it's kind of like a really nice first-line antibiotic with not a lot of bad things, but it can cause allergic reaction. So it can cause blood dichriasis, crystalluria, which is probably the most common secondary side effect above allergic reaction, which is just crystals in urine. I'm not exactly sure why it creates crystals in urine. I believe it has to do with the amount of sulfur you're intaking, as the name implies, and that likes to form more crystals with urea in urine when it passes through your kidneys. so that it just kind of makes crystals, which is not fun to have. It hurts. Hematuria and thyroid hypoplasia. But these are all very, very low occurrence things, like not super common. Okay, penicillins, which is this guy right here. Penicillins include things like amoxicillin, ampicillin, penicillin G, penicillin V, so on and so forth. Cephalosporins, we just talked about that. What they all look like is they all have this one thing which is called a central beta-lactam ring. That's an important one. Make sure you guys know it. What is it? Well, it's basically this ring that's really constrained. So carbon, carbon's okay with being straight. It's okay with being at an angle. It's really not okay if you try to make it into a circle, especially a really tight circle like a square. And so it is unhappy with this situation. This really constrained ring does not want to exist like this. So if it gets an opportunity to not exist like this, it'll react right away to not be in that shape anymore, which makes it really effective for us to use in bacteria because they have a little region, the cross-linking of NAN units, that this will react with, and it will basically destabilize their cell wall. So it'll react with the cross-linking for the peptidoglycans, and if you aren't able to properly cross-link them, the cells just lyse because now they don't have the structural rigidity they need to hold themselves in place because they're constantly getting reacted with this. It's very effective against gram-positives, as you can imagine, because gram-positives have a bunch of peptidoglycans and highly ordered structure on the outside of their cells. Whereas gram-negatives have it in between, and they don't really care as much about it, but it does still work somewhat on them. It is considered bactericidal, so if you lyse a cell, it kills it. So yeah, works really well. Aside from one of the more common side effects of just causing diarrhea, because you're messing with the natural flora of the natural flora in your intestines, it'll cause things like allergic reactions, hives, anaphylactic shock, respiratory distress. So we'll start closing up pathways. But aside from that, you can take a huge amount of it and not get any bad side effects. Like, it's pretty hard to get a bad thing with this. Now, most of you guys probably taken at some point in time amoxicillin or ampicillin or penicillin with clavulanic acid, which is a very similar molecule, and it was its own antibiotic at one point. But we don't actually give people clavulanic acid to be an antibiotic anymore. So why do we put clavulanic acid with penicillin together? Does anybody know the reason why? Anybody have a crazy guess? So what ends up happening is clavulanic acid is very similar in structure. It doesn't work as well, but it binds really well to penicillin resistance genes. So the penicillin resistance proteins that are being produced, it'll bind to them and get in their way so that the real antibiotic can start working really effectively. So even if you have resistance to penicillin, if clavulanic acid's also added there, then it starts working to prevent it from interfering with the penicillin. Really handy, super fun, cool little mechanism. Okay, so tetracyclines, how do they work? So tetracyclines include things like tetracycline, oxytetracycline, doxycycline, which is the really commonly prescribed one, and minocycline. As the name implies, it's tetra for 1, 2, 3, 4, cycline rings, 1, 2, 3, 4 rings. So that's kind of the easy way to tell when something's a tetracycline. How does it work? It binds to the 30S ribosomal subunit of bacteria. So we don't have a 30S ribosomal subunit as humans, we have different ones. So it doesn't bind to ours, so it's relatively safe for us. And what it does is it basically interferes with the bonding between the tRNA codon, the tRNA and the mRNA codon. So basically, as you're trying to put the correct amino acid in and line it up with the mRNA, it just prevents that binding and just kind of gets in the way. So all it's doing is just causing problems. And so when that happens, the bacteria has to have a much slower time making proteins than it needs. So it slows everything down. That's why it's considered bacteriostatic. It is considered broad spectrum. And the major side effects with this are nausea and diarrhea. Other weird ones are discoloration of growth plates and unerupted teeth. So if we give this to children, it will cause this weird banding on their teeth for their adult teeth, which we didn't know about when we first made it. So we don't give it to kids anymore unless we really, really have to, but that is a side effect that's gonna happen. It can also cause damage to kidney and liver, which is really bad. And another one that's kind of wacky, so it's not so much a side effect, but it does have an unusual property, which is it chelates in milk or calcium. So if you're taking like calcium supplements, So if you have a patient that is an older woman that's taking supplemental calcium because they have bone loss, bone density loss, you gotta warn them, hey, look, you can't take it with this one because it'll start chelating in your stomach and you won't get it absorbed properly. And so that's an important kind of like, hey, watch out for this. So there's another one. Or also don't take it with your morning cereal because there you go. Okay. The next one is fluoroquinolones. Fluoroquinolones, what they do is they inhibit topoisomerase 2 and 4, which are needed for DNA replication and transcription. So they just kind of like pull DNA apart, so that's their whole job. And the way it does this, there's a bunch of different examples in here, which is ciprofloxacin, levaquin, enrofloxacin. Ciprofloxacin is a really, really common one. But what it does is it kind of looks like the structure of adenosine. So, and I know it doesn't quite look like this, but they've all got the same structure. They've got this double ring structure with the fluorine sticking out, and then some other ring structure to the left, and then something right below on this nitrogen. And it doesn't quite look like this unless you squint really hard, but a lot of these structures are in there, which is the nitrogen right here, this smaller ring structure, and this large tail sticking out. That's what these all kind of have in common. And what it does is it kind of binds in that place, and it basically prevents it from unspooling DNA in an appropriate manner. So if you can't replicate your DNA, if you can't make new proteins, then you can't live, so you die. So it's considered bactericidal. It is considered a broad spectrum antibiotic, so that means it works on a lot of different things. Its side effects, though, are kind of weird. So one, erosion of the cartilage and weight-bearing joints of children, which is bad because we don't want to give children arthritis, but that can be a thing that happens. So I don't give it to children anymore. Another one that's actually really important for prescriptions that people don't think about is it causes tendon rupture, especially where tendons like to join to the bone. And the reason why this is important is that this is an antibiotic you want to be very careful if you have an athlete that does a lot of high contact or high impact sports. And we've actually seen this in the sports world several times where somebody will be prescribed this and they don't know that, and they'll do like a little juke move, and then, you know, the tendon in their leg snaps. Well, it snaps not because that tendon was weak from just natural deformities, it's weak because we gave them that antibiotic that caused that erosion. And so that's actually something you have to be really careful of. If you have an athlete that's very active and has a really high impact sport, you can be like, hey, take it chill for a while, or maybe choose a different antibiotic unless that antibiotic is really required, and then just like make sure that they're aware of that risk. Vomiting and rash can be things, so allergic responses. Abnormal liver tests is another one. So if you have a patient that's having liver failure, you want to be careful when you give that to them because you can mess up the way, especially if you're monitoring their liver enzymes repeatedly. It might cause some overlapping issues, nausea and diarrhea, things like that. So you can see that this one's works really well, but it does have some really consequential issues given the correct circumstances. So you have to be careful with some of these antibiotic prescriptions. Another one is macrolides. Macrolides are these very large ring structures. They're really, really cool. The big common ones are erythromycin and Z-Pak. Z-Pak's really, really commonly prescribed. I think it's one of the most prescribed antibiotics in America, which the Z in Z-Pak stands for azithromycin, which is this guy right here. What they do is very similar to the tetracyclines. They bind to the 50S ribosomal subunit and they inhibit peptide elongation. So they basically do something very similar, they just bind to the other part of it. So instead of binding to the 30S, they bind to the 50S. It's very effective against most gram-positives and some gram-negatives. It's not considered quite broad spectrum, but it does work on a good chunk of them. It is bacteriostatic, so it just slows down growth. It doesn't kill things. And it does have some side effects. The first one's rather minor, which is gastrointestinal upset. The other one, which is kind of big, is hepatic injury, so it can cause liver damage. For the most part, because the liver is regenerative, we're not too concerned about this unless the patient's under liver failure or has hepatitis. That can cause a lot of things, problems. But generally speaking, it's one of the safer antibiotics, so it's really hard to get any serious problems with it. Okay, last antibiotic that we're going to be talking about today is aminoglycosides, which are one of my favorites. They're very interesting. The two examples, one of them is gentamicin, which we talked about earlier, and streptomycin, which is a really cool and interesting antibiotic. If you guys are ever interested in the history of how he discovered them. Streptomycin has a really interesting story behind it and kind of the issues with giving credit in science and how we kind of discover antibiotics. It's really a fascinating story, but we won't get into that right now. It is most effective against gram-negatives, and how it works is it binds to the 30S ribosomal subunit and inhibits protein synthesis. You might be thinking, hey Josh, we just had one that did that, right? That was tetracycline. This one does it in a slightly different way, and it's actually really neat. So this is a ribosome, 50S, 30S subunit. Then in here, we have the green guy, which is our mRNA running through here, and then we're bringing in tRNA to bind with the mRNA to bring in the appropriate nucleotide, the appropriate amino acid. So everything going right, this will come in, interlock with the correct spot on the mRNA and goes, oh, yep, this is the right one, and put that amino acid in there in the chain and then go on to the next one. But what it does, aminoglycosides, they bind to this region here and they basically stick out into here and they allow codon misreads. And what a codon misread does is it says, hey, just put in any amino acid in there. It doesn't matter as long as it's like mostly right. And so now it starts shoving in the wrong amino acids. Well, you really only have to mess up an amino acid in the middle with one amino acid to make that protein just not functional. It just doesn't work at all. Sometimes it gets really bad and it will just fall out of solution. It will no longer be soluble and it'll just collapse on itself. And when you have proteins that are coming out of solution and precipitating, especially inside a cell, they want to stick to something because they don't want to just be floating. So they'll tend to glom themselves onto the cell wall and cause cell lysis. So it's actually a really serious problem if you start putting in the wrong amino acid. Even if it only happens once every protein, well, that means 0 proteins are coming out that are working properly, potentially. So it's actually a really cool little antibiotic for that one fact that it causes those misreads. So it's considered bactericidal. It's very, very effective. Side effects, super serious. One of them is allergic reactions, and it can cause severe allergic reactions, as in you can die rather quickly from just a little bit of it. Nausea, doesn't seem like a big deal, but loss of hearing because it damages the auditory nerve in ears, which is a problem, and renal damage. And I mean, it starts damaging kidneys quite heavily. So this is a good thing to think about when you've got a patient who's maybe got diabetes. People with diabetes, that's not very well controlled, they tend to get a lot of infections. If they get an infection, you want to treat it really quickly because their immune systems and their healing ability is just not going to be good. So you're like, hey, I'll give you this antibiotic because it works really well. But now if somebody's got diabetes, the likelihood of them having messed up kidneys is already pretty high, and now you give them a drug that gets stuck in kidneys and causes their kidneys to fail. Now you cause them to fail and lose their kidneys, and now they don't have kidneys anymore. And it's just one thing after another. So it's an example of a small little problem catalyzing into a huge problem. So you have to be very careful with these kinds of things when you think about antibiotics. You might be like, yeah, that's fine. We can treat this aggressively, but maybe you shouldn't because that might have consequences. You never know. Okay. And I'm not saying that's the case for everyone who's diabetic. It's just an example scenario. And a lot of things had to go wrong with that patient to kind of get to that scenario. So how do we test how effective an antibiotic is? Well, one of the things we do is we want to basically create an even lawn of growth of the bacteria on a plate. And so what we'll be doing is a 4-way streak, where we'll streak the bacteria on this plate, zigzag back and forth, up and down, side to side, up and down again, so we have an even area where bacteria will evenly coat the plate. We don't want to have any gaps in here because gaps will make it more difficult to read the results, because the results are a zone of inhibition, an area where bacteria do not grow. We call this a Kirby-Bauer antimicrobial sensitivity test. Make sure you know that name. Kirby-Bauer, it's fine, it's a short version, Kirby-Bauer test. And the way we do this is we use a type of media called Mueller-Hinton. Mueller-Hinton's the media, Kirby-Bauer is the test. Very important, remember those. And we will dispense these antibiotic discs. Each of these discs have a little letter code on them. E, erythromycin, CYP, ciprofloxacin, S, sulfa, G, gentamicin, what have you. But not all of them are the same. So for example, sulfonamide's like TET. Why? That's just the code that they use. So be aware that you have to know what the codes mean. You can't just guess. And then on the bottom, they have a number. The number corresponds to how many micrograms are embedded in that disc, so that you can calculate kind of how effective it is. And then we incubate them. So the way we dispense them on them is we actually have a little machine that does it for you guys. You just put the machine over your plate, press down, and it clips off all the discs in there and drops them in place for you, which is super handy. And the discs are already labeled. You put it in the incubator, and then you guys take it out, and then you guys just measure the diameter where the zone of inhibition is, the area where the bacteria do not grow. So just straight line across there you go. If you don't have a perfectly straight line, so right example over here, you don't have a straight line because there's a lot of no growth right here. We can just take the radius here and double it, and now we have the answer. So there you go. Now, the real question though is what constitutes a zone of inhibition? It has to be complete inhibition, no bacterial growth whatsoever. So if you see right here, we've got some colonies that are penetrating into here. They were not killed off by this antibiotic, so that means that we just measured the closest colony. So you can see that this is considered resistant because it's growing right up to the disc. Another example is right here. So if you notice, there is kind of a ring right around this area right here. It's a pretty big ring. But then if you look a little bit in, there's kind of like a fainter ring right here. And there's a little bit of light bacterial growth right here. That light bacterial growth is just the bacteria that are kind of struggling to survive, but they're still able to survive. So we consider that not inhibited. So we have to go right here where there's zero growth. So from here to here, that would be the zone of inhibition, from here to here. We wouldn't measure from here to here, because there is bacteria growing in this area right here. So just where there is zero growth. Okay. Now, how do we measure this? Well, we have to do it for each specific antibiotic, and this is actually like a little picture. This really long chart, and actually, oddly enough, the size of font in there is very, very similar to what you'll find in the box, is the list of all the antibiotics and then the zones and different ones for different bacteria. It's exactly like this. It's super tiny and really, really hard to read. But basically, each of these antibiotics has their own number, and the reason why is because it depends on the bacteria that you're testing against. So for example, in here, you can see for gatafloxacin, you have one number for Enterobacteriaceae and Staphylococcus species, but then you have another number for gonorrhea or another number for Streptococcus pneumoniae. So each of these affects those in different numbers, and so the resistant number is different for them. The other thing is different antibiotics have different rates at which they spread in media. So some are really soluble and they like to spread out far. A great example of this is penicillin. One that you can actually see in this image here. If you guys look all the way down to here, to the gemafloxacin, you can see that it's considered susceptible, the bacteria is considered susceptible to this antibiotic if it has a zone of inhibition that's greater than 23 millimeters. But it's considered resistant if it has a zone that's 19 millimeters. But if you look all the way up here to Etropetum, if it has a zone of inhibition of 17 millimeters, then that means it's susceptible. And its zone of where it's resistant is only 15 millimeters. So it's got the exact opposite. So it's, it's got a very small ring for susceptibility. So if you compared it to another antibiotic, it wouldn't use the same zone because one antibiotic's susceptible number is another antibiotic's resistance. So keep that in mind. Okay, so each of these has to have their own number that we have to measure, and sometimes it's specific to an organism. So keep that in mind for these. What you guys will do, because you guys don't have two antibiotics, you'll just choose whatever your unknown organism was, and you're going to streak that on a plate. It doesn't matter if you have gram-positive or gram-negative. It doesn't matter, whatever your unknown organism is. If you don't have an unknown organism that got isolated, we will make copies for you guys, and that way you guys can streak your own bacteria, but just keep that in mind that we will have those available. The source organism will be your unknown. You'll just do that four-way streak that we talked about. Try to minimize any kinds of gaps. You will then bring it up to the instructor desk and use the little machine to clip off all the little discs that you have. You guys will only have like 6 discs. You won't have one that looks like this, but. And that's the basic way that you'll do that one. You will also do something called an e-test, which stands for epistolometer. And basically all it is a little strip of, it can either be nitrocellulose fiber or polyvinyl plastic sheeting fibers, and we embed them with different types of antibiotics at different concentrations, And each of these concentrations will tell us basically how effective it is at killing this particular bacteria. And so we can just basically keep looking up this chart and see where the bacteria stop growing, and we can tell what the minimum inhibitory concentration is, where it starts killing off the bacteria significantly. So here, for this one, you can see for DLX, we can look and we can see it basically starts at around 0.006, where you start having no growth around the strip. The trick with the strip, though, is you want to make sure it's kind of in the center of the plate. If it's too skewed, sometimes it gets a little weird in how it grows and disperses. So just try to get it right in the center of the plate. It's actually really easy to do. You'll get one of these plates to do per group of 4. We'll give you an unknown organism to do, or it's not going to be unknown, it'll be known organism. So we'll give you one of those organisms to do, and so you'll streak it out just like you did for the Kirby-Bauer tests. You'll do the same streak pattern, but then you'll place these strips on your plate, so your secondary plate. So you'll just have one of these e-test strips for your plate. And it's really, really easy. So you'll just incubate it and you'll just look for that zone of inhibition. Simple test, very, very easy to do. Now for you guys will do your test results also for gram-negative on Monday. No, sorry, not Monday for you guys. Tuesday. So you guys will review the results. You'll do this antibiotic test, and then you guys will leave, and then you'll come back and do the rest of the antibiotics on Thursday. Now, does anybody have any questions on the antibiotic stuff that I covered so far? Everything make sense? Yes? Yes? Cool. All right, so we're going to talk about our cool science thing of the week, which I am really excited about. So you guys probably won't be because it's kind of complicated, even though the title is really simple. The title is Programmable Protein Delivery with Bacterial Contractile Injection System. What does that mean? That's kind of vague. So if you guys remember, way back at the beginning of the semester, we talked about a glowing bacteria that caused infections that glowed in the wounds of soldiers. Does anybody remember what bacteria that was? No? You remember a guess? Wild guess? Okay, that was Photorhabdus species. And it turns out that Photorhabdus species, even the one that they were talking about specifically in that cool science thing of the week, Photorhabdus asymbiotica, has a method of communicating with other bacterial cells and its host cells, which are nematodes, using this thing, protein delivery system, this guy right here, which if you just look at, it kind of looks like a T-phage virus. That's cool. And it does look quite a bit like this. It's kind of in a lot of ways very similar, but structurally different. And so what it does is it has this little guy right here, and inside of it, it will package separate proteins that it can send out, and it'll send us those signaling molecules to another bacteria that it wants to communicate with, so within the same species, or in the nematodes, to give the nematodes a signal to do something else, because they live in the guts of nematodes, or in one of these cases, these are toxin proteins, and they'll kill off insect cells, which is kind of cool. So they can not only alter other bacterial cells, but they can also send these protein things into eukaryotic cells. And so we're like, well, let's see if we can abuse this system. So the way it works is you've got this whole exon setup of genes, and you've got, or codon region of genes, and you got a whole bunch of proteins. Each of these proteins are structural proteins that help build up this little spaceship guy, along with a binding tip area. And then you got some other proteins here that are involved with packaging and binding of your target protein so that it goes inside of this guy. And lastly, on the very end of this stretch, you have this guy and this guy, which are toxins. And in here you have some middle bits, these orange ones, which are regulatory genes, and we don't know what they do, but they definitely are there. You don't necessarily need them, but they are kind of important. We're not exactly sure how important or what they do, which is not very helpful, I know. But they are important. And so the thing is, we know that these exist, but we don't know a whole lot about them. So these guys are like, we're just going to figure out what they all do and use them to infect other cells. Not really infect. It's not an infection system. It's protein delivery system. So they're like, hey, let's see if we can take this. So they took this gene out. they copied it and clipped it into E. coli. Because E. coli is a lot easier for us to manipulate, and we can do a lot of things with E. coli. Put it in E. coli, we isolated these little delivery vehicles and ran a negative stain and then ran it through an SEM. So we got a micrograph of it. And so you can see electron micrograph. Oh, these are right, these are the right size. This is what they should normally look like in real life. And these are the ones that we got from E. coli. We ran out of gel. Yep, it's making the appropriate size things. We're like, hey, let's put a tag on these guys. And we know that these end regions are sticky and like to bind to a insect cell receptor. So we have this insect cell line. So we put this little fluorescent protein on here, stuck them on here, and turns out, yeah, it binds these insect cells, and the receptor part out here is green, so it makes a nice green glow. And then we're like, hey, can we put proteins inside that glow? So they're like, oh, okay, let's do that. So first off, they did one with this guy, which is the PDP1, which is a toxin, and they put it into insect cells, so normal insect cells with nothing in it, and then normal insect cells with this protein and this toxin in it, and it killed off all the protein cells. And then they're like, let's mutate it a little bit, and when you mutate it, it doesn't work. And then they're like, let's mutate this packaging protein, and when you mutate the packaging protein, it doesn't package in there, so the cells survive too. So that was their controls. And then they're like, hey, let's stick these fluorescent proteins in here. And they put in CFP, Korean zinc fingers, and lo and behold, they were able to get them in there and glow as well. And then they were like, hey, can we put DNA in there? So they put DNA, loaded it up in there, and we put GFP expression plasmids in there, and they were able to program and infect them into these. These guys are, yeah, these are the same S19 cells, so the same insect cell lines. So they were able to bind and put that one in there and transform them, which is really handy. All right, so let's see what else we can do. And so they're like, hey, one of the problems is that this, to get stuff loaded into here, we definitely know that we need this region at minimum, because people have already done research on that, and they were like, this region, very minimum, we absolutely need. But this protein's really long, it's got a bunch of this other junk. How much of this junk can we clip off and still get that protein loaded in there? So they ran an experiment where they did this truncation deletion, they cut off a whole bunch, and it turns out you just need this little tiny bit from PDP1, and you don't need any of the other one to get this guy to load in, which is really handy. So they were able to get this system and figure out, okay, we only need these two little bits to package it in here, handy. But now we need to get it to bind to things regularly. And so here's where the problem comes in. It does look, if you squint really hard, like those phages, right? And we already know that phages are really good at binding very specifically to bacteria. But the reality is that these are not phages, so they only bind to one specific thing, and they're not quite like this. We don't know exactly how they work. So what these guys did is they were like, let's just pretend that they're exactly like them, and we're just gonna mutate them the same way we'd mutate those tail fibers, which is not an amazing thing, but they basically kind of lucked out here. And they're like, we're gonna use a computer program to help us guess what these parts do and guess where we can cut here. So this is what this protein, actually, this is what this protein looks like normally. So this part attaches up here, and this is the part that binds to stuff. And they were like, okay, this part that binds to stuff, how much of this can we clip off? And it turns out we can clip off a lot and still get this little tube deal remaining. So the tail part that sticks, the tail part, you can cut off a lot of it and still have that tubular tail part intact. And they were like, okay, now we can add stuff to this linker. So they added the end of this thing called adenovirus, which is a cold virus for humans, adenovirus 5, and they added this thing called knob, which binds to human cells, and they put it in human cultures, and what do you know? Binds into there, and we get a whole bunch of binding. Okay, it works. Well, now we need to test to see if we have appropriate controls, so we'll make some amino acid deletions that we know inactivate this, and now it doesn't work. Controls verify that working is doing it in the intended way. Then they're like, let's take another one. We use DARP-N, put it in there, and you can see DARP-N looks wacky in comparison because this guy just kind of sits out here. DARP N kind of folds up into itself and creates that weird flower structure. Okay, works. Transfers in there. Now we can see it glowing. Excellent. Let's put some non-targeting binding residues on there. So we're basically putting in amino acids that don't work properly, so you need those to work. And we lose that function. So that means we got there. Sorry, got a lab assistant, and they are just letting me know certain things are happening. Sorry, I don't mean to have my phone there, but they're off doing their own thing. So we know we can target this to human cells, and we can, oh my goodness, I'm just gonna turn it off now. If they have a problem, they'll just call me, or the police, one of the two. Okay, so they're like, okay, now what can we do? So we were like, hey, how big of a stuff can we jam inside of here? Because the inside of that tube, the really big tube right here, that's hollow. So this tube right here is for this tail. So to give you some scale, this is 50 times the diameter of this. So it's big. So we're like, how big is it? So we start putting in huge proteins, and we start loading in a bunch of things. And one of the interesting ones they did was they did zinc fingers, which are rather large proteins. And zinc fingers are dimers, and so you can actually just separate them out, but they should be stuck together as dimers to do their functionality. So what they did is they had these zinc fingers. They put one zinc finger, the left side, in one of these injector things, and they put the other zinc finger in another one. They mixed them together and put them in the cell culture, and when you got both of them together, you got a mutation that caused that fluorescence, because those two zinc fingers cause a very specific mutation. We got a whole bunch of that fluorescence, and we noticed that there was a ton of mutations. But if you only put one zinc finger in, it doesn't work. Controls, very important, especially because this experiment is gigantic, as you can tell. So the next thing, they were like, let's see if we can do this in, how specific is this? Let's do it in a bunch of different cell types. So we're gonna see if we can get these guys to inject a protein into these guys and see if we can get GFP being produced. And we wanna make sure that these are binding the appropriate tags only. So the only time that you should see fluorescence is if the right tag is on the cell surface, and these guys can bind that tag. So we did the experiment. So each of these cells, each of these little squares that you can see here is its own separate experiment where they're putting different combinations in together and mixing them. And the only time you should see fluorescence is when HA is matched up with the anti-HA. And so you can see it lines up perfectly there. There are no other colorations anywhere else. And of course, no tag and no antibody, you shouldn't see anything because that should be blank, because nothing should be glowing. But you can see it lines up perfectly, and there's no spread fluorescence, which is good. So that means that it's really, really specific. It only goes where it's supposed to go, and it doesn't go anywhere else that it's not supposed to go, which is actually really important because specificity is really key, especially when you're doing targeting of human cells. Oops, and I guess I skipped over that. So there's one more picture in this, which is actually really cool. So the next thing you do is like, oh yeah. So we can do this in cell culture, that's great. We can do it in human cells, we can do it in mouse cells, we can do it in insect cells, we can do it, all these things. We can put DNA in there, we can put proteins in there. Well, can we do it in a living thing? Well, they got some mice. These mice are special because they have a gene in them that we've made, which is called LOXP T tomato, which is a wonderful name. And the way this works is LOXP is a region where we can clip and flip something around. So it's a recombinase sequence. And T tomato is a fluorescent protein that makes bright red. But it only works if it's in the correct orientation. So what we did is we made a bunch of these mice, and then we injected into them these protein things, and inside the protein things we had the recombinase for LOXP, which is called CRE, in there. We injected into the mice, we then looked at the mice's brains, and they started having red glowing areas only when they had the appropriate binding regions on the delivery system. So we could make their brains glow a bright fluorescent red, which I didn't want to show you. Some people get squeamish about seeing mice brains, even though it's like 90% black, but they're still brains. So, but there you go. So we can do it inside things. Why is this important? I know I've talked about this forever. We can give you, as a drug treatment, proteins. We have lots of antibody treatments. There's tons of short peptide treatments that we have now. Insulin, for example. We've got tons of different treatments for like migraines and stuff. But we have problems getting small proteins into cells. Getting it into the cell is really hard. This system gets us to put proteins into cells, and not only just puts it into cells, it does it with high specificity and with a whole bunch of different cell types, which makes it super convenient as a drug delivery system. So this could be the future of drugs, where we're like, hey, we've got this now protein. Boy, you have this cancer and it only responds to this one drug type. Well, great, we can just load this one drug type into this cancer and then inject it only into the cancer cells because it's highly specific. That's what the future looks like. So all you have to take away from this is we stole this thing from Photorhabdus and loaded up our own proteins in it, and now we can make it bind whatever we want and inject the proteins into those cells consistently. Okay, I know that was a really complicated thing. This is actually like literally cutting edge research. This was released on the 29th of this month. So I've had all of 2 days to read this, but it's really, really cool. It's like the cutting edge of cutting edge stuff. And it will probably be a very big future thing, these protein loading systems. So keep that in mind. All right, if you guys have any questions, come by and see me. I'll let you guys go now. Sorry for talking so long. Have a wonderful weekend, and I will see you guys next week.